Okay, so tonight we're going to be studying for your organic molecule test. So our number one is, in terms of science, what does the term organic mean? And organic is anything that it has carbon and is a living thing, or was a living thing. So we see the word organic often in grocery stores, and organic means that there's no other chemicals put into it. There's no pesticides, no fertilizers, it's just carbon. And so that's what this chapter is about, is our carbon-based molecules, and you also might hear it be called organic molecules. Okay, what is it about carbon's atomic structure that makes it the building block of life? So the carbon atom has 4 valence electrons. So if you have carbon, it has these 4 valence electrons, and it just, because it has 4, it has this uncanny ability to pretty much bond with anything. And so it could bond with a hydrogen, oxygen, a phosphorus, sulfur, and many other things, but in living things, those are the only things that it'll bond with. But it's the 4 valence electrons. Remember, valence meaning outside shell. All right, the different types of organic molecules we have are cats, love, pea, nuts. It's a way that I remember it. We have carbohydrates, and lipids, proteins, and nucleic acids. All right, and these carbon molecules are going to be at their smallest molecule, which a molecule is a covalently bonded pair of atoms, or maybe 6 atoms. Just one molecule is going to be our monomer. So here in D, you can see this is our monomer, where this long chain is our polymer. So if we were just going to describe that, we'd say our monomer is our one molecule, and a polymer would be many molecules connected together. And so in class we referred to this maybe being the train car, and then all of them hooked together being the train. All right, first we're going to talk about carbohydrates here. The 3 elements that make up carbohydrates are carbon. All of them are going to have carbon, right? And then we're going to have hydrogen. And our last one is oxygen. And just these 3 are going to make every carbohydrate that we have. So your bread, your candy, any grains, so like rice, all your vegetables, all your fruits, all of them are made of only 3 atoms: carbon, hydrogen, and oxygen. Okay, so what is the ratio of these elements? So whenever there is 1 carbon, there are 2 hydrogens for that carbon and only 1 oxygen. So our most common carbohydrate is glucose, and there are 6. So if there's always 2 hydrogens for every 1 carbon, that would give us 12 hydrogens and 6 oxygens. If there was 8 carbons, then we would have double that 8 because of this. So we'd have 16 hydrogens and 8 oxygens. And so these carbohydrates are built off of this 1 to 2 to 1 ratio. Okay? All right, so these are the 3 major polysaccharides that we kind of looked at in biology. So down here, and then this one is our main monosaccharide, excuse me. So glucose is what all of these are going to break down into. So glucose is what our cells use for energy. And so every other kind of carbohydrate we eat, our saliva, our stomach acid, breaks it down in our stomach to make glucose. Fructose is a really similar compound to glucose, and it's found in fruit. And it can be changed in our body to glucose. So glucose, where is it found? It's in the It's the monomer of carbohydrates. And it is what the cell uses for energy. Okay. Starch is something that we get in potatoes and French fries and anything that has a root. Okay, so if it is something from under the ground, it's going to contain starch. And this is just really long strands of glucose. And these are going to be found, they're really big polymers, polysaccharides of glucose. And they are going to be, when we eat them, we get lots of energy. But sometimes that's not a good thing, because if you're not burning that energy, then that energy or those glucose molecules are going to turn into lipids. All right, glycogen, this is if our body doesn't use some of the carbohydrates that we eat. Glycogen is, we can store it in our muscles. So I'm kind of writing what these, I should be writing it over here, but where it's found, it's in our muscles in animals. And this is exchanged into glycogen in our liver, and it is carbohydrates that are stored in the muscle, and they can give us real quick energy. So if you've ever heard of carb loading, like for the night before a big race or volleyball game or basketball game, that's what they were doing. They were eating a whole bunch of carbohydrates. Our body turns it into glycogen, stores it on our muscles, and then when we are in our activity the next day, it stores for about 24 to 48 hours, and then it's able to be real quick energy for our muscles, and we don't have to be eating in order to get it. All right, cellulose, this is the most common carbohydrate in the entire earth, and this is like wood. Okay, and so this is a cell or plant structure, and like a cell wall, but cellulose in particular is wood. It's very hydrophilic, so it doesn't absorb water, and that's why wood floats. Cellulose, we cannot digest it, so animals cannot digest. Well, at least humans. Some animals can, like cows, they eat grass and has cellulose in it, but large amounts of cellulose, like wood, animals can't digest it. And so we would have to have lots of enzymes and bacteria to help us break that down. All right, next one, lipids. The three main types of lipids are going to be our triglycerides. And triglycerides are going to be fats and oils from plants and animals, and they're going to be stored energy for us. When we eat them, they travel through our bloodstream, and they get stored as fat, so that when we run out of carbohydrates, that we have something as a backup for energy. Our next one is phospholipids. And phospholipids are going to be for our cell membrane structure. And our last one is our cholesterol or steroids. Okay, anything that's a hormone. And these are for regulation in our body for a whole bunch of different things. So like testosterone, estrogen, those are kind of hormones that regulate, female and male organelles and all sorts of different things. And they also regulate body temperature. Hormones are just such a huge part. Cholesterol is a specific type of lipid that we can find in our bloodstream. And we have to keep our cholesterol very low to prevent heart disease and stuff. Just to remind you what these look like, triglycerides are going to be one glycerol head with 3 fatty acid chains. Our phospholipid is going to be one glycerol head with 2 fatty acid chains, and they're going to stack up together because of their polarity and be a really good barrier for our cell membrane. And then most steroids, they are a carbon ring or a couple of carbon rings kind of put together, so they kind of stack on top of each other like this. And they're also a lipid. They have those fatty acids that kind of wrap up around. So there's 2 types of triglycerides, saturated and unsaturated. I kind of mentioned them already. Saturated fatty acids are carbon-to-carbon single bonds, and they are going to be our animal fat, and they're typically solid. They're not going to be liquid. Where unsaturated acids, they are going to be our plant oils, and they are going to be liquid. The reason they call this saturated is it's saturated with hydrogen. And so it talks about that a little bit in your book, but the word saturated means saturated in hydrogen. All right, fill in the table below. We want to state solid, liquid, gas at room temperature, commonly found in which type of organisms, and types of bond connecting the atoms. So saturated fats are going to be solid. I guess I kind of wrote that up there, but that's okay. Commonly found in animals. And they're going to be covalent compounds. Covalent bond, a sharing of electrons. Where unsaturated fats are going to be liquid. And they're commonly found in plants. And they are also covalent bonds. Okay, what's the structure below? Label all the parts of the structure, including the polar and nonpolar regions. So this structure is a phospholipid. I know this because it only has 2 fatty acid chains. So these are the fatty acid chains. And this is the glycerol head. Okay, and so our glycerol is our polar head, and our fatty acids are nonpolar. And all this means is that it's going to be a really good barrier for cell structure, for cell membranes, because it's going to keep the water out of the middle. All right, nucleic acid is our next one. Fill in the table of nucleic acids. So our monomers are going to be nucleotides, and there are 5. We have the A's that go with the T's, and the C's that go with the G's, and then in RNA, we don't have the T, we have the U. So we have adenine, thymine, uracil, cytosine, and guanine. And remember, the guanine can also go with the cytosine, and the thymine also can go with the adenine. Polymers are RNA and DNA. And DNA really is the storage of genetic information, and it holds that inside of the nucleus, where the RNA's function is really to transmit the genetic information, and it carries it outside of the nucleus and to the ribosome to form a protein. So it transmits the genetic info, but they both have to do with genetic information. All right, so let's talk a little bit about the nucleotides. So the arrangement of nucleotides determines the kind of protein that's going to be created, because depending on how these are ordered, they're going to bring in different amino acids in the ribosome. Okay, so circled an entire nucleotide. So we have the phosphate, the 5-carbon sugar, and the nitrogen base. So this would be one nucleotide. and it's attached to another nucleotide over here. And then how many nucleotides are shown in the DNA segment pictured? So we have 1, 2, 3, 4, 5, 6, 7, 8. And the 3 parts are the phosphate group, which is right here. And then we have the 5-carbon sugar. And then we have our last one is the nitrogen base. And this is why, so in our last slide here, all of these fats are made up of carbon, hydrogen, and oxygen, just like our carbohydrates are made up of carbon, hydrogen, and oxygen. And that's because they're going to transmit back and forth. You know, we're going to eat extra glucose someday, and it's going to get put into a lipid, and then the lipid, we're going to run out of glucose some days, and we're going to have to change that lipid back into a carbohydrate. So they're the same 3 elements, where a nucleic acid has phosphorus and nitrogen. So we have the phosphorus here, the carbon, hydrogen, and oxygen in our sugar, right? And then the nitrogen in our nitrogen base. So there's the elements of our nucleic acid. All right. And it says fill in the DNA molecule above using the letters. So we'd put P's by all of our phosphorus. These are all phosphorus. This is our backbone. And then our sugars, our 5-carbon sugars. And then we have adenine, guanine, thymine, cytosine. Okay, so if I put an A here, we know this has to be a T. Put a C here, this has to be a G. If I put a G here, this has to be a C. And if I put a T here, this has to be an A. And that's that base pairing rule. Base pairing rule. That talks about those A's going with T's and the C's going with the G's. All right. And then, of course, one strand of this, if we were just going to look at one side of this, this would just be an RNA strand. So 2 strands are DNA, 1 strand is RNA, and then in DNA, these nucleotides are connected by what we call a hydrogen bond. And so we get a hydrogen bond instead of just all these covalent bonds. All of the rest of these are covalent bonds that are sharing between. All right, let's talk about our last one, proteins. So fill in the table. So we have amino acids. Remember that there are 20 of them, 9 of them that we have to eat. So you can't produce the proteins that you need unless you are eating food. So our polymer is going to be our polypeptide chain. And then it's going to be folded a whole bunch, and we're going to finally make our protein. But the first polymer that comes out is our polypeptide chain. And the function of a protein is to repair our muscles when we eat it. And then when it's just in our body, it's really controlling everything from our heartbeat to the color of our hair. It's carrying out the instructions of the DNA. So it carries out instructions of DNA. And that's why it's just so important. Okay, so how many different amino acids are there? So that's 20. Fill in the blank. The blank of an amino acid determines the type of protein made. Even one incorrect amino acid placement can change the protein's structure and function. And this is the R group. Okay, so the R group is, so all amino acids have this, we call this an amino group. and this is a carboxyl group. This is something we mentioned. You wouldn't have to know that on the test, but these are the two groups that are in every amino acid, and then this R group is going to be what changes throughout. Okay, so what's the molecule pictured below? This is amino acid. What type of bond forms between amino acids? And this is covalent. If you haven't noticed, all of these carbon molecules are all covalently bonded together. They're all sharing electrons because carbon covalently bonds with everything. Label the molecules, parts A and C. Okay, so this is the R group, and then the amino group and the carboxyl group. Write another term. What's another term for protein? So this could be like I think this is also down here for an enzyme or a catalyst. So circle the group on the structure, circle the group on the structure pictured below that is different for every one of these molecules. That's the R group. I accidentally circled those earlier. Draw on the molecule pictured the two arrows where the bonds would be. Where these bonds would be. So right here, these are peptide bonds. And so this is one amino acid, this is in the next amino acid, this is the next amino acid, and this is all happening in the ribosome. So we have our nucleus here with our nuclear pores, and the DNA is going to make RNA, and the RNA is going to come out, and we're going to have all these little strings of nucleotides, no T's, only U's. And we're going to have a ribosome that it finds. It's going to lay down in it. And there's going to be these little structures that match up perfectly with the nucleotides on the RNA, and they're going to carry amino acids. And we're going to learn all about this later. And these amino acids are going to start covalently bonding together in the ribosome. And this is our polypeptide. And when it's done, it'll fold all these different ways, and eventually we'll make a protein. Okay, so proteins are made in the ribosome with instructions from our DNA that turns into RNA. That goes, finds the ribosome, and then amino acids all connect together with peptide bonds, and then we finally make our protein. Okay, so our next part, one really important function of proteins is the fact that they are enzymes inside of our body. So we definitely can eat proteins, and they carry out processes in our body, but they also are enzymes. And so enzymes are going to be catalysts. So if you remember, that speeds up a chemical reaction inside our body. So every time a chemical reaction needs to take place in our body, whether we're using water, or we're breaking down our food, anything, breaking down carbohydrates, an enzyme is what's going to house that chemical reaction, or it's going to be what holds that molecule and breaks it down, causing a chemical reaction to happen. So another term for an enzyme is a protein or a catalyst. And why are they enzymes important? Because chemical reactions take place there. And there's lots of chemical reactions that take place in our body. Okay, so it says just review the characteristics of an enzyme. So they're very specific. We talked about this with the lock and key model. Lock and key would be like, if you drink milk, there's the lactose carbohydrate in there, and in your stomach there's the lactase enzyme. So ASE means enzyme, OSE means sugar, and so only lactose can hook to that lactase. That's it. So they're very, very, very specific. If you drink maltose in a milkshake, or if you have apple, fructose won't break down with that lactase enzyme. So they're very specific. They're reusable, so After the chemical reaction is over, another lactose can just fit right in there. And they're very sensitive, so they can very easily stop functioning with temperature or pH, and we call that denaturing, which I think is on the next slide, but that's okay. Whoops. Let me start over there. Denaturing. Okay, so when an enzyme is denatured, it stops working and starts breaking apart, and a couple of things that can cause a protein to be denatured would be like if you got a really high fever, or if for some reason your pH in your body changes. All right, so there it is, what causes an enzyme to no longer work. So this could be a change in pH or temperature, and we call that denaturing. Okay. All right, write an explanation of the process below. Identify all components of A through E. Okay, so B, C, and D, all of these are going to be our enzymes. Well, it's just one enzyme, and this is how it's moving across. So we have right here is our substrate, and this is just a molecule that needs to be broken down. It could be a carbohydrate like lactose. It could be bacteria that is our white blood cells, our immune systems are connecting onto. So it could be a big range of things because proteins are so diverse that there's lots of things. But so A would be our substrate, B would be our enzyme. This right here would be called our active site. This is where it has to be really specific, okay? So not just anything is going to fit in there, okay? This is where the chemical reaction is taking place. And we've talked about activation energy. So when this takes place, since it's a catalyst, the activation energy is decreased, and so it doesn't take as long for the reaction to happen. And then this last one is our products. This is two new parts of whatever it is, or it's a bacteria that's been broken down, and it's used to be your substrate, that was one, and it's been broken apart to two. It also can start out as two, and an enzyme can actually put something together. So enzymes are doing lots of great things in your body, keeping you alive. And anyway, I hope this helps you with your test tomorrow. So continue to study, and we'll see you tomorrow.